Welcome to this short review of humoral immunity. It is one of the major branches of the adaptive immune system. Hi, I'm Christopher, and in this video, we'll review antibodies and the cells that make them. We'll focus on how B cells become plasma cells and what antibodies actually do, and why the humoral immunity is so important for protection against threats outside of the body's cells. The word humoral comes from the Latin word humor, meaning liquid or fluid. Historically, the word was connected to the old idea of the four humors of the body. In modern immunology, we still use it to describe the antibody-mediated branch of adaptive immunity because antibodies are found in bodily fluids such as blood, mucus, secretions, and lymph. The main cell that we think about with humoral immunity is the B cell. B cells develop from precursors in the bone marrow, although the B originally comes from the bursa of Fabricius, an immune organ in birds where the cells were first studied. For mammals, though, B for bone marrow is still a very useful mnemonic. Here's a quick overview. A naive B cell recognizes a specific antigen through its B cell receptor. With the right signals, including help from T cells in many responses, that B cell becomes activated. It can then differentiate into a plasma cell, which is essentially an antibody-secreting powerhouse. And it can become then a memory B cell that helps the body respond faster the next time the antigen appears. In casual science conversations, people sometimes say, antibodies kill bugs. But that's not quite right. Antibodies do not usually kill pathogens by themselves. Instead, they bind to specific targets and help the immune system control or eliminate those threats in several major ways. First, antibodies can neutralize pathogens or toxins, meaning that they block attachment, entry, or function. From the body's perspective, neutralization can almost be as good as killing because the pathogen or toxin can no longer interact effectively with host cells. Secondly, antibodies opsonize microbes. Opsonization is like putting a bright label on a pathogen that says, "Eat this." Cells such as macrophages and neutrophils can recognize antibody-coated microbes more efficiently and phagocytize them. Third, antibodies can help activate complement, one of the soluble mediator systems we discussed with innate immunity. This can amplify inflammation, increase opsonization, and in some cases, contribute to microbial lysis. Fourth, antibodies can cause agglutination. Because antibodies have multiple binding sites, they can cross-link microbes or particulates together. This clumping can limit spread and make clearance easier. Thus, the big idea is this: antibodies are not usually tiny assassins. They are more like blockers, flags, connectors, and amplifiers that help the rest of the immune system do its job. Most mammals produce 5 major classes of antibodies: IgM, IgG, IgA, IgE, and IgD. A quick comparative note: other species can have additional or different antibody classes. Birds, reptiles, and amphibians have IgY, while sharks use the antibody class IgNAR. Insects, including bees, do not make antibodies in the vertebral sense. For this video, we'll focus on the 5 major mammalian antibody classes. IgM is usually the first antibody class produced in a primary response. It is large, works well, works early, and is especially good at activating complement. IgG is the major antibody class in the blood and many tissues. It is the one we often discuss with systemic protection, vaccine response, and long-term antibody-mediated immunity. IgA is important at mucosal services, including the respiratory, gastrointestinal, and urogenital tracts. It helps protect surfaces where many pathogens first try to enter the body. IgE is important in defense against parasites, especially helminths and arthropods, but it also plays a major role in allergy and anaphylaxis. IgD is mainly a receptor on naive B cells. Its full role isn't clearly understood compared to the other antibody classes, but it's still part of the B cell biology. The key point is that antibody class matters. Different antibody classes help place humoral immunity in the right location for the right job. Now let's walk through how a naive B cell becomes part of an effective humoral response. A naive B cell boasts thousands of identical B cell receptors. That is, it can only bind one specific antigen. When that B cell receptor does bind antigen, that B cell can internalize and process the antigen. In many important antibody responses, especially those to protein antigens, the B cell then presents the antigen to the helper T cell. If the B cell receives the correct T cell help, including co-stimulatory signals and cytokines, it becomes fully activated. Once activated, B cells undergo clonal expansion. That means that antigen-specific B cells divide and create more cells with the same basic antigen recognition. Some activated B cells become plasma cells and secrete large amounts of antibody. Others become memory B cells, which persist and allow the immune system to respond more rapidly and effectively if the same antigen is encountered again. In germinal centers, B cells can undergo class switching and affinity maturation. That helps the body improve the quality and function of the antibody response over time. Thus, humoral immunity is not just make antibody. It is a carefully coordinated process that turns rare antigen-specific B cells into powerhouse plasma cells, memory cells, and improved antibody responses. Let's bring this all together. Humoral immunity is the antibody-mediated branch of the adaptive immune system. It is especially important against extracellular threats, including many bacteria, toxins, viral particles, parasites, all before they enter host cells. B cells recognize specific antigen. Plasma cells produce antibodies by the truckload. Antibodies help neutralize, opsonize, activate complement, and agglutinate targets. Memory B cells help make the next response faster and stronger. So when you think of humoral immunity, think B cells, plasma cells, antibodies, extracellular protection, and memory. Thanks for watching. I'm Christopher Lee, and what you understand today helps you protect patients tomorrow. And I'll see you in the next video.